Hello everyone, my name is AJ Keefe, and in this presentation I'll be covering the paper Mitochondrial Dysfunction Induces Dendritic Loss via eIF2 alpha phosphorylation, published in the Journal of Cell Biology. So I want to extend a special thanks to the authors for their contribution to this field of research. And for this presentation, I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. If you have any questions at all, please comment below, and I hope you enjoy the video. This paper revolves around better understanding the eIF2 alpha, or just eIF2a, integrated stress response signaling pathway. At its core, this pathway involves integrating multiple different stress signaling pathways that all converge on the phosphorylation of eIF2a, which results in the inhibition of translation. So let's start with eIF2a. This protein is a critical component of the ribosomal pre-initiation complex. When eIF2a is charged with GTP, it is able to bind the initiator tRNA that recognizes methionine. And during the formation of this ribosomal pre-initiation complex, eIF2a uses its GTP, converting it to GDP. And in order for eIF2a to be used again, it needs to exchange GDP for GTP. So eIF2b, a different protein, eIF2b charges eIF2a, allowing it to form a new pre-initiation complex for translation. However, and this is big, eIF2a can be phosphorylated at serine 51. And this phosphorylation prevents eIF2b from binding and swapping the GDP for GTP on eIF2a. In other words, eIF2a phosphorylation locks the protein into an inactive conformation, effectively stalling translation across the cell because its GTP cannot be regenerated. Interestingly, not all translation is stopped by this. In fact, translation of alternative downstream open reading frame transcripts are actually promoted by the phosphorylation of eIF2a. One very well studied alternative reading frame that is upregulated following eIF2a phosphorylation is ATF4. It's a transcription factor that effectively turns the stress response into an actual transcriptional response. So any pathway that converges on the phosphorylation of eIF2a is going to be stalling translation, and it's going to be known as part of the integrated stress response. And it comprises these four main kinases. You will notice that each kinase that targets eIF2a is itself coming from a unique trigger. PKR, for example, is activated by a unique trigger, and that is the viral response. And let's see, GCN2 over here is activated by amino acid deprivation. And PERK, which is going to be important for this paper, is activated by ER stress. Normally, it's activated by ER stress. And so this PERK eIF2a ATF signaling pathway is eventually shut off by a phosphatase that cleaves the phosphate off of eIF2a, allowing translation to resume as it normally would. In mammalian cells, this phosphatase is GAD34, and it is a phosphatase that is upregulated by ATF4. So that might sound paradoxical, but it is a negative feedback loop to restore basal conditions. So this GAD34 is actually a subunit of PP1 that gives it specificity towards eIF2a to cleave its phosphate. So this phosphatase in Drosophila is called DPPP1R15. And for the purposes of this paper, it's very important that you remember that DPPP1R15, which I'll be repeating 100 times, is functioning as the eIF2a phosphatase that inhibits the integrated stress response and restores translation by cleaving the phosphate off of eIF2a. Step 1 is to establish a model of mitochondrial dysfunction, a starting condition, and neuronal dysfunction, a result that can be measured. So they use these Drosophila peripheral sensory neurons that, when overexpressing PREL, experience mitochondrial dysfunction, culminating in the severe loss of dendrites. And you can see this pretty clearly in the images. The wild-type neurons show expansive dendritic arborization and a well-connected mitochondrial reticulum. In PREL mutants, indicated by the PREL1 condition, the dendrites are devastated, and the mitochondria are highly abnormal and clustered. Likewise, in the PREL overexpressing conditions, the dendrites and the mitochondria are again very abnormal. The first thought was that ATP is probably downregulated. Because you have these highly dysfunctional and fragmented mitochondria, it's reasonable to believe that their ATP levels are probably not the same. So they use these fancy ATP biosensors that bind ATP, and upon doing so, they emit fluorescence. The biosensors are called A-teams, or in the graphics they're called ATNL. The non-ATP binding control is called ATRK. So in this top right figure, they show that PREL overexpression did not significantly change the FRET to CFP ratio. The FRET to CFP ratio is a standardized measurement of ATP concentration using these biosensors. So we don't need to get into the specifics, but the FRET CFP ratio is simply a relative measurement of ATP concentrations using this ATNL ATP biosensor. And what they observed was that ATP concentrations remain mostly stable during development. We see early L2, early L3, and late L3. These are developmental stages of the larva of a Drosophila. And you see the neurons from these stages. You see the ATP concentrations in these neurons. And what this suggests is that the ATP concentrations are mostly not significantly different from their controls. The researchers then observe ATP concentrations following inhibition of glycolysis or OXPHOS. So AM, I think, is glycolysis, OM is OXPHOS inhibition. What they observe is that the amount of ATP in wild-type cells, control cells indicated by the black line, drops off rapidly following inhibition of glycolysis or oxidative phosphorylation. PREL overexpression, on the other hand, dropped off much more gradually following inhibition of glycolysis or oxidative phosphorylation. So in other words, PREL overexpressing cells are able to maintain their ATP concentrations because they simply are not using nearly as much of it. Because when you inhibit the production of ATP, the concentrations remain relatively stable over time. As a researcher seeing these results for the first time, you might think to yourself, what signaling pathways might downregulate ATP use in order to maintain standard ATP concentrations, as we observed up here? Well, stopping translation by eIF2a would be a pretty effective method in slowing ATP use because translation itself is a very expensive cellular process. Also, dropping some dendrites and reducing membrane surface area, where many of the expensive ion pumps and synapses are found, would also help a cell maintain ATP concentrations. So the next step is to identify what underlying signaling pathways connect the dots. So we have a loss of dendrites, we have mitochondrial dysfunction, and we have conserved ATP concentrations at the L3 larval stage of development. And I should note that all of the experiments from now on are using these L3 larval stage development because ATP concentrations are conserved, and that gets rid of the confounding variable. And so this is the phenotype. Now, disturbing what pathways, modulating what pathways will mitigate this phenotype. And they tested several of them, such as the P53 dominant negative, didn't change the length of the dendrites. BSK dominant negative, this is an inhibitor of the J and K stress signaling pathway, didn't change anything. They also did a BAC, didn't change anything. The one gene that they found to significantly alter dendritic loss was the DPPP1R15 protein, a known eIF2a phosphatase. So the first step after this discovery was to confirm that it was indeed functioning as an eIF2a phosphatase. So they placed DPPP1R15 under the inducible Gal4 promoter, UAS. And when they induced DPPP1R15 expression using this system, they found that eIF2a phosphorylation to drop precipitously. So in this second lane, where DPPP1R15 was induced, they found that there was almost no eIF2a phosphorylation compared to the first lane. So this is under normal conditions. When they artificially upregulated DPPP1R15, they found that the amount of phosphorylated eIF2a was almost absent. They then took this inducible system as well as a second DPPP1R overexpressing model, and they found that when DPPP1R15 was upregulated by either mechanism, it significantly increased the total length of dendrites. So this was just one figure showing this. There was a couple others, and the mCherry-CAX is a control for the number of transcripts that were in there. So, but there are a few other variants of this experiment, including some nice images. But the fundamental discovery was that an eIF2a phosphatase, DPPP1R15, partially restored the amount of dendritic arborization and total length of dendrites. And it also occurred in the absence of any change in mitochondrial morphology. So the mitochondria still looked highly abnormal when the DPPP1R15 was upregulated, but it was instead likely an event downstream of eIF2a phosphorylation that was being altered by the upregulation of DPPP1R15. So their next goal was to identify the kinase that was placing these phosphorylations onto eIF2a in the first place. And they knocked down various known eIF2a kinases to see which one restored dendritic arborization. And they found that PERK was the culprit that was phosphorylating eIF2a, because knocking down PERK right here, restored dendritic length. Now, exactly how dysfunctional mitochondria are upregulating the activity of an ER localized stress sensor was not investigated, but that's definitely a central question. So the researchers also observed that IR1, another ER stress sensor, was upregulated in the nucleus. So PREL overexpression induces mitochondrial dysfunction, eIF2a phosphorylation through PERK, and also the transcription through IR1. And interfering with this pathway by either downregulating PERK right here, or by upregulating an eIF2a phosphatase right here, restore dendritic length for the most part. So at this point, we've been discussing a single model, and we're talking about what happens when a cell overexpresses PREL in their mitochondria. But is this really applicable to other forms of mitochondrial dysfunction? To test this, the authors made 3 different models of mitochondrial dysfunction. The OPA1 overexpression, a protein of the inner mitochondrial membrane that modulates fusion. OPA1 lacking its GTPase and fusion ability in this condition. And TTM50 overexpression, which is part of the mitochondrial translocon. Now, I am entirely unsure why these proteins were chosen to induce mitochondrial dysfunction, but I do not have any experience with Drosophila, so maybe it's a fly thing. I'm not exactly sure. But they used OPA1 overexpression to induce mitochondrial overfusion, so a hyperfused mitochondrial reticular network. And the OPA1 K273A point mutation was used to form a hyperfragmented mitochondrial network because it lacks the ability to fuse mitochondria. And the TTM50 overexpression, I'm not actually sure about, but I guess overexpressing part of the mitochondrial translocon causes dysfunction somehow. Regardless, what they found was that across the board, these proteins cause loss of dendrites, and that DPPP1R15 restored dendritic length. So it's really as simple as that. So this ultimately suggests that eIF2a phosphorylation is common to multiple forms of mitochondrial dysfunction, because we can see that in the the PPP1R15 overexpressing condition, when that is present, right, in the purple columns, the dendritic length is significantly restored in each condition of mitochondrial dysfunction. So if you induce mitochondrial dysfunction in any of these three conditions, overexpressing the eIF2a phosphatase restored dendritic length. This research ultimately suggests that eIF2a phosphorylation, primarily by PERK, may be fundamental to mitochondrial dysfunction-induced dendritic loss, even in the absence of ATP depletion. So this means that ATP depletion following mitochondrial dysfunction is not a direct cause of mitochondrial loss, because many people kind of assume that dysfunctional mitochondria would struggle with ATP generation, and this would cause a general energetic deficiency and cell death, but that does not appear to be the entire case. It may be that mitochondrial dysfunction downregulates ATP use, and it does this through eIF2a phosphorylation and by downregulating its dendrites, but ATP is still largely conserved. The second big finding was that components of the UPR, the unfolded protein response, this is an ER stress signaling pathway. This was found to underlie these signaling networks that downregulate dendrites during mitochondrial dysfunction. So my first question, was how in the world are these mitochondria activating PERK? Because PERK appears to be the central kinase that is phosphorylating eIF2a, but PERK is an ER stress sensor. So what is the bridge between these two organelles? And in line with this question, how is IR1 activated? Normally, IR1 is activated by ER misfolding by a splicing event, a non-canonical splicing event. So how does eIF2a phosphorylation actually lead to dendritic loss? We are left completely in the dark on that point. Presumably, it must be some protein that is upregulated by an alternative reading frame, perhaps, because remember, eIF2a stalls translation except for those downstream open reading frames. And there might be a dendrite destroying protein that is translated by downstream open reading frame, or perhaps the loss of translation itself maybe starves the dendrites of some critical proteins. And what general function does eIF2a phosphorylation actually serve? Why is it upregulated following mitochondrial dysfunction? So is it possibly to help conserve ATP by stalling translation and thus helping a cell conserve ATP? And this isn't obvious at first, but is dendritic loss a good thing? Because these cells we're dealing with have dysfunctional mitochondria that are only able to maintain ATP concentrations because ATP is being conserved and used less. Perhaps ATP isn't being used as much because the cell got rid of a bunch of its dendrites. So dendrites are expensive little organelles, so maybe it's a good thing they're downregulated, because otherwise, if they kept their dendrites alive, maybe a cell might catastrophically self-destruct due to ATP depletion. Does ATF4 is the canonical downstream transcription factor of eIF2a, play any role here? So I was stunned the authors didn't actually investigate this. ATF4 is a transcription factor activated by eIF2a phosphorylation because it's only transcribed by alternative open reading frame translation. In fact, a recent paper by Kuros et al., published just 3 months before this paper, found that ATF4 was the primary transcription factor that communicated the mitochondrial unfolded protein response. And additionally, ATF4 was eIF2a phosphorylation dependent in this paper. So both these papers, I think, complement each other very nicely, because this Quiros paper suggests that mitochondrial dysfunction in the form of unfolded proteins signals through eIF2a and ATF4. And the one big caveat, however, was that in the Quiros paper they eliminated PERK as the responsible kinase. In fact, they couldn't even find the kinase responsible for triggering eIF2a phosphorylation. But nevertheless, these two papers together suggest very strongly that eIF2a and ATF4 is a central signaling network following mitochondrial dysfunction. And so my last question, pretty basic: can these findings be replicated in human neurons using models of human mitochondrial dysfunction? And I think that's where this research will go next, and I'm very excited to see where it goes from there on. So thanks for listening. Please post any questions you have. I only went through a couple of the figures of this paper, so there's probably some things that might be missing. So if you have any questions, please ask, and it might spur some interesting conversation. And thank you again to the authors for this amazing paper, and thanks for watching.